right. First of all, I need to figure out which one to use first. Back. All right, good morning, everyone. My name is Fan Yi. I'm a senior scientist from Center for Diagnostic Innovation at CHOP. So thank you for the opportunity to present my work on the potential plasma biomarker for STXBP1 related disorder via targeted proteomic approach. So I guess the title already gave out. The target here is the STXBP1 protein in plasma. And then thanks for the nice introduction from Dr. Boland. So basically my background is already set. One disclaimer though, the aptamer we're gonna use is actually not developed by us. It's actually from StomaLogic. I think you already showed in one of the slides. We contacted them, they can actually sell the individual aptamer instead of like as a kit for everything. All right, so for this, well, I'm a bioanalytical scientist by training, so I hope I'm not the only one in this community. So today my talk is going to be heavily focused on the method development portion. And then, so dive into the background a little bit first for STXBP1. I think for this community, we already know it's a rare disease, and we know it's caused by a pathogenic variant in this gene, STXBP1 gene. And then the clinical phenotype is kind of like you have a really wide range. So, and often this is in most of the children with STXBP1 related disorder, the pathogenic variant is a de novo mutation, so you can get limited information from family history. And also, a lot of speakers has talked about the haploinsufficiency issue, so all of them contributes to elevated difficulty in diagnosis. So currently, I think there's no approved disease-specific treatment yet. And then most of the treatment is focusing on symptom management. So that almost kind of like necessitates a early diagnosis. Speaking of diagnosis, I think the primary tool right now is genetic testing. Of course, there's additional tests like EEG and MRI to augment that. So there is kind of like challenges in this process. I think a lot of speakers already mentioned. First of all, the clinical phenotype is very wide, and then also you have the de novo mutation. Then with the existing mutation, you have the variant of unknown significance. So all of them contributes to sometimes inconclusive decision. And then moreover, for genetic testing, it's not really effective in therapeutic monitoring purpose. So at CDI, we were kind of like, we were joking around, like we have a tunnel vision. You give us a task, and you receive the result. So when the ENDD team contacted us, we were given the task to develop a quantitative LC-MS assay for STXBP1 in plasma or CSF. But of course, plasma would be the preferred media here because it's less intrusive and it's more available. So the initial goal is to use this as a treatment endpoint monitoring. And then eventually, if this assay works well, we were trying to develop this into a full-on complementary diagnostic tool. So as I just mentioned, I'm a bioanalytical scientist by training. So today my talk is going to be focused on these three major assay framework here. So first I want to introduce you to the technology LC-MS and also how we select the unique peptide for STXBP1. And then finally some sample property issue that we encountered during our method development. First of all, so to the audience who is unfamiliar with the concept, LC-MS stands for liquid chromatography tandem mass spectrometry. So all the assay workflow starts with sample preparation. In this case, we use tryptic digestion. And then once you have the sample ready, you inject into the LC. The liquid chromatography, basically, to simplify it, is a separation technology. So basically, you have a column packed with reverse phase material. And then that's what you call a stationary phase. And then your peptide will go through that stationary phase, have some sort of interaction. And then your mobile phase, which is some sort of a solution, will carry your peptide forward. And then hopefully by the time your peptide comes off the column, it has a well separation from other interference in this case. So once your LC eluents comes off, now you officially enter the mass spec portion. So in this case, I'm using a triple-quadruple mass spectrometry. The ESI source is where the analyte get turned into this gas phase ion. So As you can see in the graph below, the LC eluent comes off the column, and by applying a high voltage and then also nebulizing gas, you are turning to a charged droplets. And then because the high temperature and high energy, the solvents start to evaporate. And then at some point, your charge density will be so high, and then it will just burst into this very defined gas phase ion, and then that's what it will be introduced into the mass spec. So after you go through the ESI source, now you are facing several molecular filter, that's what we call it. So as the name implies, triple quadruple, so it's 3 sets of 4 metal poles arranged in parallel. So you apply a RF or DC value to it to create an electric force. So that's the way you can, so basically you can tell the mass spec, okay, and to select the specific value you input, and then the filter will basically adjust it, and then only select the species that you tell the mass spec to select. So, okay, yeah, so each molecule, each peptide in this case, you know the exact mass, and you know how many charges is associated with it. So now you have a what we call a mass to charge ratio, m/z. So that's the number you're going to tell the mass spec to select. And then in the MS1, which is the Q1 in this case, you will select the precursor m/z. And then once the selection is finished, the charged particle will enter collision cell, which is Q2 in this case. Then go through a process called collision-induced dissociation, CID, and then that will produce a bunch of product from the peptides. And then again, for the product, you will have another set of m/z value, and then by telling the mass spec, okay, you need to select this now. In the Q3, which is MS2 here, you will select the product ion. So by establishing a transition from the precursor to product, you tell the mass spec, this is your target, and then only those targets will reach the final detector and generate a response. That's how the mass spec can afford its selectivity in this case, and then gives you the reassurance that you are actually monitoring whatever you intended to. So for LC-MS work, actually the final data output is actually a 2-dimensional data. So most commonly people will see the chromatogram, which you can see is the red one. And then basically at different retention time, you will have a peak, and then that peak corresponds to the analyte you are monitoring. But for LC-MS, now you can extract that peak, and then you can tell what exactly is underneath it. So as you can see on the blue graph, for example, on the 3rd peak, there's actually 2 species that's co-eluting. So by using the mass spec data, you will be able to tell which one is which. So that's why this platform is so powerful and is so selective. So you heard me talk about a lot of input. So basically what you need to tell the mass spec is what species you want to monitoring. So that's the next step, is to select a good target. So in this case, for STXBP1, we need to generate unique peptides. So basically, a lot of the work will be done in silicon first on computer. So through this algorithm, you will basically, it all boils down to your final peptide selection. You have the characteristic of, it must be unique. That's kind of like the most important thing for this protein in the species, in this case in human. And also you want to make sure the peptide is somewhat stable and also ionizable. And finally, you would hope for a more conservative peptide, which means it should present in all the isoform, hopefully, and also it's the lack of any sort of polymorphism or post-translational modification. So on the right, I'm showing you the 7 peptide we initially selected, and then the green one actually is the one that we are focusing on right now. And then remember, the top portion is all happening on computer, and then finally at the bottom, you will customer synthesize the peptide standard, and then you try to inject into the mass spec and LC. So here I'm just showing you a really simplified version. Within a 2-minute gradient, you can easily separate all 7 peptide and then see their response. And another thing, I think Dr. Boland showed one of the chromatogram. For each peptide, we are monitoring more than 4 different product. That's another way we can confirm that the peptide we are monitoring is actually what we are monitoring. So now, finally, it comes to the sample preparation issue. That's most of the time, that's the most challenging part for this whole workflow. So because of the, for this type of application, most of the time the protein is low abundance. And then also make it more complicated is that the STXBP1, we all know it kind of interact with the other protein in the snare complexes, and also it can be membrane encapsulated. So it can be decompartmentalized in different location in the sample. So that's where we spend a lot of time trying to figure out, okay, how do we recover STXBP1 from plasma in this case? So not to overwhelm you with information here, just trying to show you like there's a lot of different ways we tried, and none of them are working very well. To speed up, so the most important thing is actually we figure out we need to use a detergent, and then we also need to make sure the method is specific to STXBP1. So during the whole process, we made a very interesting discovery. First thing is there is a negative correlation between the centrifugation speed of how you prepare plasma to the peptide concentration you recover. And then second is the free store cycle stability. Usually when you look at those data, you would think free store cycle stability, you should expect the concentration of the peptide protein start to drop as you go through more free store cycle. However, we saw it reverse. So this brings us to two questions. Is this possible that STXBP1 is related to platelet count? And then second is kind of like the protein is encapsulated by a membrane. Well, in the end, you realize this is actually the same problem because platelet is a membrane structure. So finally, we got to the point like we decided to use aptamer, which in this case is a SOMAmer from SomaLogic. And then also because of the platelet, we developed a specific platelet marker, which is called CD41 protein. So basically, we quantify that as well for normalization. So this is the final assay workflow. To save time, I'm just going to quickly say, in the end, we just report out two concentrations. One is the STXBP1 concentration. The other is CD41 concentration. And then this assay is still in the infancy phase, so we haven't done a ton of validation. But through the couple accuracy and the precision run, I think the performance is pretty well. You can see for both standard curve, you have close to 1 R-square value, and also for the level of QCs, it's within 15% bias. And the more importantly, if you look at the bottom table, if you just look at the straight up STXBP1 concentration, there's a 4.5 times difference. However, after normalization, you bring them back close to 95% of each other, which is very good. And then finally, we analyzed 50 plasma samples from combined brain. And 23 of them are control, and 27 of them are diagnosed patients. However, because it's like a biobank, we don't have any control over the quality of the sample, like how they stored, how it was transported, and how the sample was prepared. So if you just look at the STXBP1 concentration straight up, it's not very significant. I mean, most of the concentration actually is like heavily focused on the bottom portion of the graph. However, once you start to normalize with CD41, you can see the stratification gets better. And then we also realized for CD41, not only you can use it as a normalization scheme, but also you want to use it as a quality control purpose. So basically, like if your normal control has no CD41 reported in it, that means that's probably not the sample we want to accept. So once you've taken out all those samples, now it's much more significant, and you can see a clear stratification between the control versus STXBP confirmed sample. And then I think the reduction is from about 1.5 to 0.45. So that's a very good start for us. And then this is the kind of like the latest data we have. So I intentionally left out the future direction because right now we're still trying to optimize our assay. We recently acquired a microflow LC stack in the CDI lab, so we're trying to optimize that so we can have a much more sensitive assay. And then in that case, we will actually be able to run mouse plasma sample from Ben Prowser's lab. So, of course, this brings a lot of questions like why is this in plasma? And then is there like what's the ratio between the free STXBP1 and then platelet-bounded STXBP1? But those are all something we want to address in the future. So that's my last slide regarding the data, and then quick shout out to all the CDI team, and then without the help from Dr. Master, CDI team is not going to happen, and then also a big shout out to the ENDD team to reach out. And finally, thanks for the foundation for the invitation, and then Combined Brain for the samples. I'll take any questions.
Yeah, thank you. Very interesting data. I was wondering, can your method, the LCMS, can it detect the difference between mouse and human or NHP?
Yes, it can. I think mouse and human, the amino acid sequence is different. So if we want to develop a test for mouse, sure, you can. I think the most challenging part is to find a homologous aptamer to actually targeting the mouse version. But I think for the mouse model they were developing, actually they're using human STXBP1. So
but I
that shouldn't be a problem.
But I thought the sequence was identical more or less. But are there some?
The protein sequence is not identical across the different species. And the same thing goes with CD41. We actually was able to develop a CD41 in mouse already. We just haven't done that for the STXBP1 yet.
Hi, thanks so much for the interesting talk. I think I have 3 questions, and maybe you can answer the one that you think would be most relevant to the group.
Sure.
One is, since you think the STXBP1 is within platelets, would platelet-rich plasma be a better sample matrix going forward? That's one. 2, if you're measuring a platelet protein, does it reflect anything in the central compartment? Like if there was some therapy that was directed at the brain, would it change that measure? And the third is, what does it say about STXBP1 function, maybe outside the brain, that it's being found in platelets?
Yeah, very good question. I may have to use one of the call for help cards to Dr. Poland. But to the first question, I think, yes, platelet-rich plasma would be the go-to matrix that we're going to use for this assay, because if you remember the graph I showed, like if you spin the platelet at a lower centrifugation speed, you get a much higher concentration from STXBP1. And we're trying to avoid using the platelet-depleted plasma. So that's the first question. And the second is, I don't really know if there's like a direct translation between the CNS to the plasma platelet, but I think that's one of the questions that we should address in the future, because I can only tell you.
It's the mouse experiment where we're trying to see if a delivery targeted to CNS can have an effect on platelet STXBP1 at low and high speeds. So we're using the mouse model to address that question.
Thank you.
Forgot about the third question, sorry.
Well, I was just asking about the function and the biology of STXBP1, that it's found, within platelets that are, cell fragments of megakaryocytes as a bone marrow.
Yeah. So that kind of relates to the.
What does that mean?
We don't think STXBP1 is expressed at low levels in platelets. It's mainly STXBP2 that is ready to rebuild.
Yeah, I actually stole this slide from you. So.
So we think that what's going on is STXBP1 somehow trapped in extracellular vesicles that are bound in the platelet. That's our current.
Oh, okay, so not really a platelet protein.
Exactly. We think that it's probably CNS. It's probably STXBP1 from the CNS and extracellular vesicles that are found in the blood. That's what our goal is. We're going to try. We've got a lot of experiments
Did
platelet-derived microparticles from other extracellular vesicles. Virtually the platelet
That was really beautiful, thanks. Did you happen to look at syntax?
Well, that's the plan. That's actually the next one.